Welcome back to Chapter 24, the transcription and post-translational processing of RNA. This is part 3. There were 2 previous parts where we spoke about what is RNA, and then in video 2 we talked about the entire process of RNA transcription. So now we're going to talk about how RNA can be modified. And so in our outline we have moved, if this part is video 1, this is video 2, and we are here in video 3. So when we think about mRNA or RNA, we know that it is a really short-lived product. And so much of the rRNA and tRNA is found in pre-RNA. When we think about prodrugs or precursor molecules, we know that it is not yet the final product. And so a pre-RNA means that it's got to be modified, whether that's chemically modified or anything else. So the mRNA transcripts are modified to create a more functional unit. So tRNA is known to be transfer RNA. And this is involved in the polymerization of proteins. And so this is how our amino acids are going to be carried into the growing peptide chain in order to create a protein. And so about a third of the RNA molecules have been covalently modified. And so our RNA is modified to form the subunits. So our RNA is ribosomal. So the ribosomal RNA encompasses the ribosome, which we will also meet in chapter 25. So pre-RNA modifications. So tRNA transcripts are covalently modified. If we look at this image up here, we can see that there are parts of the rRNA, the ribosomal RNA, and 2 different tRNA constructs. This is all a single transcript. And so what we see is 2 tRNAs. These are 2 different tRNAs, and then 3 different components of the rRNA, all within the same transcript. And so we need to covalently modify many of the bases, and we have to remove it from this component. So in this other graphic, we can see that different RNases, so in this case we have RNase P and RNase E, and so these 2, RNA-D, come along and create modifications. So the other thing is we have methylguanosines, isopentyl adenosine. These are covalent molecules or chemical changes to RNA components. What we'll see in chapter 26, 5, in the next chapter, what we're going to observe is that these covalent modifications allow us to determine and differentiate between the different transfer RNA. So our types of covalent modifications. These allow us to create the specificity. So what you can see over here are some of the chemically modified bases. So we can increase by adding methyl groups, or we can alter the base structure. So we have an isopentyl group, or we've changed out to create an inositate subgroup. All of these compounds, it's not necessarily important to memorize their structures, but it's important to note that these are permanent chemical changes. So a covalent bond is a permanent bond, and so we've altered these in such a way that they are stable in their new components. So for the next several minutes, I want to just wrap this up by thinking about what happens in eukaryotic cells. So prokaryotic cells are bacteria, eukaryotic cells come from eukaryotes, which are multicelled organisms, and that encompasses things like humans and dogs and fish. So as with replication, the higher the order system, the more complicated it is. So we have multiple RNA polymerases, and so they have additional RNA polymerases and some organelles. So if you remember, one of the things that distinguishes a eukaryotic cell is the ability to have organelles. Organelles are small components within a cell. So in this case, many of theirs are isolated to the nucleus or in the mitochondrion or in the chloroplast. These two are for specific cell types. So RNA is transcribed in the nucleus of the cell and then transported out of the nucleus for protein translation. If you remember when we talked about the end of or right after termination of prokaryotic mRNA transcripts, we said that the bacterial mRNA, once it extends out of the RNA polymerase, the translation machinery, the ability to make proteins, will immediately bind. That is not possible in eukaryotic cells due to the localization of the transcription, which is in the nucleus, and the translation, which is not in the nucleus. So there are three main polymerases. So in this case, we have polymerases that do different things. Pol-1 transcribes large RNA molecules. Pol-2 transcribes structural genes, and Pol-3 transcribes small RNA genes. So the difference between Pol-2 and Pol-3 has to do with the length and the type of information that is encoded within that gene. So the E. coli, or the eukaryotic polymerase, if we look at the structure, it's much larger than the E. coli variant, but the overall mechanism is similar. We see these kinks, which we observed in the bacterial or the prokaryotic observation. We also see an open complex, which you can see here, which is still on the order of this prokaryotic, the prokaryotic cells. And so what we see is the ability to create mRNA in the very similar mechanisms where the DNA must be separated after the promoter regions. So initiation is not the most complex of this component, but it is quite complex when we think about how this works. And so in this case, we have 4 different binding areas. We have a TATA box, which is upstream. We then have the TFI-ID. So when we think about all of these factors over here, they bind to different parts. So if we think about the sigma subunit that assembled the prokaryotic RNA polymerization holoenzyme, we now have multiple players. And so these multiple players bind to different parts of these sequences with different specificity. So there are 5 transcription factors required for RNA polymerase II to transcribe. So many class II genes, protein encoding, contain a TATA box located 19 to 27 base pairs upstream. So to count this, we consider the ATG as 1. This means that it starts somewhere between -19 and -27 bases upstream of the start codon. And so these protein encoding factors, the TATA box is important for the assembly of this. So transcription factors. This is really initiated by the TFI-ID binding to a TATA box or other initiation sequences. So when we look at these genes over here, we can see that they're transcriptional. So here's our start codon. But all of these are promoters. So there are a lot more promoters. Some of the upstream mediators can actually interact with the polymerase. So we can control how much of the mRNA is made by some of these upstream components. Once this is composed and the open complex is formed, then we can go forward and transcribe mRNA. So enhancement, if we think about something that is enhanced, it is made stronger. So our fully assembled polymerase can be enhanced by what's called an activator. So we see our activators over here. And you can think of these kind of like the PEP squad. I know, I've gone to Texas on you all. But in this case, we think about an activator as something that enhances the response. So we are going to have activators and we're going to have things that can turn off the response. And we're going to meet both of those in chapter 6. Once we really understand how protein can be made, we want to think about how a cell can control how much protein is found at any given time. So in this case, we have our polymerase down here. These smaller green circles are many of the different transcription factors that have to bind. So we also have a nucleosome over here on the left. And so this nucleosome has to be removed or pushed aside in order for the DNA to have access to the, or for the RNA polymerase to have access to the DNA. So the elongation of transcription occurs somewhat similarly. The termination also has the ability to create these hairpins, and so eukaryotic genes also have a large poly-A tail. So the transcripts have to leave the nuclei for this to occur. The increased poly-A tail increases the stability. This basically gives time for some of the ends to be removed prior to translation, which occurs, again, outside of the nucleus. So these 250 poly-A tails, which are added by the poly-A polymerase to the end of the mRNA transcript, if we add 250, somewhere on the order of 50 to 100 are lost before it departs the nuclear pore. So this is part of the 3' UTR, which is a new acronym that we haven't met yet. So UTR stands for the untranslated region. So these are things that are outside of the gene of interest. So we've talked about the start codon and the stop codon. This is on the end after the stop codon. And so in this case, we have a relatively large, roughly 250 nucleotides, untranslated region. And so a lot of that's going to be lost prior to the departure from the nucleus. So mRNA processing is somewhat more substantial in eukaryotic cells. A lot of this has to do with the nucleus. So both the 5' and the 3' ends are modified. mRNA can be excised depending on whether or not if it has introns and exons. And all of these modifications, their entire goal is to increase the stability of the mRNA transcript. So over here, we're showing a 5' cap. So this essentially is going to add a inverted base, the 7-methylguanosine, to the 5' end. This is going to stabilize the mRNA, and it's also going to be used in the ability to know once we move into translation, which is the ability to convert RNA into protein. This is going to help the eukaryotic cells know that this needs to be translated. We do not see 5' caps in prokaryotic cells, or very rarely, if at all. But the reason we don't need a 5' cap is due to the fact that the bacterial transcription and translation machinery occurs at the same time. So mRNA splicing, this uses the ability to recompose or to alter the exons. So when we think about alternative splicing, this is the ability to make a variety of proteins from the same gene. So one thing you can notice is that if we look at this first one, we see a protein that contains 5 exons. The next one contains just 4. It's missing 3. And this one contains 4, but is missing exon 3. So the thing that you can't do is necessarily create a protein where we have 1, 3, 2, 4. We can't alter the order of these. They do need to go in the order of which we observe them. So the introns are the part of the sequence that are removed. Those can control other proteins. They can control RNA fragments that could be utilized. They can also control other types of enhancement molecules. But the exons are part of the sequence that are present in the mature protein. And so this increased protein diversity allows you to selectively express the parts of a protein that are necessary in different cell types. So if we see a type of a protein found in the heart, it might be different than that protein observed on the skin or in neurons. So the spliceosome. This is a pretty large set of proteins. It's a multi-subunit complex that removes the introns and stitches together the exons. So the mRNA fragment, in this case, contained the introns, and then they are removed. There are other instances where the introns can be never converted into mRNA. And so these require SNRPs, which are small nuclear RNAs, and this complex associates and disassociates for each removal. So it's important to really think about what the size of the loop tells you. This loop, or the intron, has to be big enough to reach around this complex. And so when it does this, there are both upper and lower limits, and so we end up with a ligated mRNA product. And so that is what will lead to protein. And in this case, it has no introns. So SNRPs, kind of a fun name. So these are small nuclear ribonucleoprotein particles, SNRPs. So one of the things that's interesting is we have SNRP RNA over here, and we have pre-mRNA, but the introns and the exons can control what parts of the protein have access to. So SNRPs can bind splice sites. They can also be used to prevent mRNA production. And so SNRPs are just one way that eukaryotic cells are able to control the production of mRNA. So this concludes the third video of the chapter 24.